All right, so for today, you guys are going to be doing this plant growth lab, but we are just going to go over talking through some of the answers. Because of stuff, we're doing them a little bit out of order because some classes have not gotten to the elephant poop lab yet. Some of them, we are still waiting for your plants to grow. So looking at this, based on the seed experiment in the poop lab, which seeds sprouted and grew? If we're looking at what we expect to happen, we should expect that the seeds that were in the fertilizer or in like the fecal matter of an animal are going to sprout and grow. And then how do we know this? The class, I think fourth period, you guys started this lab. If you want, you can peek at your seeds. Otherwise, how do we know if a plant grows? Right? We can see it sprouting. So you're going to write that evidence here. And what happens to cells when living things like the seeds grow? Well, those different cells are going to grow, and as they get bigger, they are going to then split. That is what actually makes things grow. So the cells will grow and then split in the process of mitosis. So if we go down to the bottom here, it says, today you will observe cells in two parts of a plant, the root and the leaf. So down below I have some images from a microscope slide of a root and some images from a leaf. Okay, so if we skip down, and it says in the diagram below, you will see plant cells taken from a sample. When the nucleus is present, we say the cell is an interphase and not preparing to divide. When the nuclear membrane breaks down and chromosomes begin to become visible as dark lines, we say the cell is in mitosis and preparing to divide. So mitosis is going to consist of specific phases that occur in a specific order. Prophase is the first phase of mitosis where chromosomes condense and become visible but are still tangled together. Metaphase involves chromosomes moving to the center of the cell lining up. Anaphase is where the chromosomes are being pulled to opposite sides of the cell. And finally, telophase is where each group of chromosomes are at either end of the cell and unravel in their previous state. And then cytokinesis is where the cell actually physically divides. So if we look down at this picture of these different cells, we can see that we have a lot of different phases going on. If we're looking at cells that kind of look like this with just that circle in the middle, that is going to be interphase. So I have a couple other cells that are an interphase. Over here, we just see their little nucleus. I'm just going to go through and circle all of the ones that are an interphase. Okay, so I went through and circled all of the ones that are an interphase. And now I'm going to grab a different color, and the next phase is prophase. So I'm going to go through and circle those ones in yellow. Those are the ones that in that nucleus, we can see they're kind of still in a circle, but we can see like lines and different things like that within that actual nucleus. So I would say that these two are also in a part of prophase. The next one we are going to look at is metaphase. In metaphase, all of the chromosomes are lining up in the actual middle of the cell. So here you can see the chromosomes are in like a little line right here. So that one would be in metaphase. I don't see any other ones that are in metaphase. The next one would be anaphase. So within this cell, after lining up in a line, some of the chromosomes got pulled to this side of the cell, and some of the chromosomes got pulled to this side of the cell. So down here, this one as well would also be an anaphase. And then our very last one is telophase cytokinesis. So this is where you can see here in anaphase, it kind of looks like two little spiders on each side. Here we see more of a condensed little circle of chromosomes on each side. This would be that last telophase cytokinesis, where we are starting to form two brand new cells. So next on this, what you are going to do is use this picture of the onion root tip. So this is the bottom part of a root. This is where a lot of mitosis is going to happen because the roots of this plant need to continuously grow to find nutrients and different things like that. So you are going to use this image to actually count up the number of cells that are in interphase, so not dividing, and then the amount of cells that are actually physically dividing, okay? So if we try to go through and answer some of these questions, so what parts of the cell can you see? In this image of the cell, I can see the nucleus. I can also see the cell walls, and not much more. So what conclusions can we make by looking at this image? Well, in the actual root of that cell, I see a lot of cells that are actually physically undergoing the process of mitosis. I see a lot of different things happening in these cells. They're not just those regular solid block nuclei. So conclusions, these cells are actively dividing. So observe the root tip and then count the total number of cells. To save time, you may utilize a similar method like the one you did in the elephant census activity. So you can estimate, if you see here, we have these individual blocks of each of the cells, so you could count the number of cells across and then the number of cells up and down and multiply those out to come up with an about number or an approximation of the amount of cells in this image, okay? Down here, you are then going to count the number of cells that are not dividing and the number of cells that are dividing and come up with a percentage. So if I would go through this image and just put a mark on all of the cells that are in the process of mitosis, I would just start over here. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. I would say probably about 19 are in some part of mitosis. So that number would go at the bottom. And then, for example, if I counted a total of 100 cells, I would take 100 minus 19, and that would be my number of not dividing cells. So I would have 81, and the percent of the total. If I got 100 cells, I would have 81% that would be dividing, and then I would have 19%, or I'm sorry, 81% that are not dividing, and 19% that are dividing. So were the cells in the sample usually dividing? Well, usually would mean the majority of them. So the majority of them are not dividing, okay? Your evidence would be looking at those different percentages. So after we look at those cells that are in the process of mitosis, we can look at the slide of these leaf cells, okay? So here, we have a little bit of a slide from some leaf cells, and the first thing that I notice is I can see the same parts of the cell. So I can see the cell wall, I can see the nucleus, those are the parts of the cell that I can see, and right off the bat I can also see that a lot less of these cells are dividing. I see 1, 2, 3, 4, 5, 6 cells maybe that are dividing. And there's a lot more cells in this picture than there were on the previous page. So I'm going to add up my different distributions and then answer these questions down here. So we're looking at these questions down here. We can see that in the root tip, we had way more cells that were dividing than in the leaf. So you can look through and look at the differences between those two samples. Down here, we are looking at what we actually know about our seeds. So with each of these, you are just describing what is happening at each of these stages, looking at this image of the different parts of a seed and answering those two questions down below. So I hope this video helps of you guys trying to figure out these different things about the plant. And yeah, make sure that when you are done with this, you turn it into the basket.